I think this is my honor to introduce the first speaker, my dear friend of very, very, very long time, although I haven't seen her almost for more than 10 years, Mariam Okhovat. So Dr. Mariam Okhovat, I met her when we were doing our undergraduate studies at University of Tehran. Then she moved to US for her PhD studies in University of Texas at Austin, working with Dr. Stephen M. Phelps. And then after finishing her PhD in integrative biology, she moved to Oregon, in Oregon Health and Science University, where first as a postdoc, as part of Dr. Lucia Carbon's lab, and then as a staff scientist, and now as an associate director of the KCVI Epigenetic Consortium. And through the years, although I know by Mariam personally, one of the main things which comes to my mind is her problem-solving skills, her amazing endeavors, whatever she's doing. So it's really an honor to have her in this symposium. And you can start, Mariam. Really happy to have you. You are muted, by the way. I'm sorry.
Oh, sorry about that. Thank you for that introduction, Aldous, and thank you for inviting me today. Let's dive right in. So a question that fascinates many biologists, including myself, is how species and organisms evolve. And in understanding this question, we often focus on phenotypes that are different between species, because understanding the mechanisms that drive these differences can provide insight into how phenotypes are shaped and how speciation and adaptations occur. But we also focus on features that are similar between species, because these features that are conserved across millions of years, despite species having diverged, can provide insight into mechanisms and processes that are vital for an organism's survival and fitness. Oftentimes, when we study these similarities and differences, we focus on the genome. And as you know, the genome is a highly complex structure. So we can study it at many different levels. People look at coding DNA, non-coding DNA, non-coding RNA, coding RNA, non-coding RNA, all kinds of, this really goes on. One of the features that hasn't really been studied much in the context of evolution, however, is a genome's conformation in the three-dimensional space. As you know, the genome is a very long molecule, and in order to fit in the very tiny nucleus, it has to go through many layers of compaction. This compaction is not random. So at the largest layer, you have chromosome territories, and then within each of these chromosome territories, you have compartments of active, inactive chromatin, known as AB compartments. Within each of these compartments, you have little modules or hubs of interaction that are called topologically associated domains. And I'm going to talk a lot about these structures in my talk today. Within these little interaction modules, you have the actual interactions themselves that are often in the form of chromatin loops, where genes and regulatory elements can come in physical contact. And then if you zoom in further, you have your classic chromatin structure, nucleosomes and all that good stuff. And then when you zoom in further, the naked DNA. The genome conformation can be studied at any of these levels, but in this talk, I'm going to focus on topologically associated domains. Topologically associated domains, or TADs, are often defined as self-interacting units of the genome, where genetic elements within the same unit interact with each other a lot more frequently than they do with elements outside of the genome. Each TAD is usually around a few hundred kb to a few megabases in size, and it can contain several genes and enhancers that interact within this space. Interactions with regions outside TADs, within neighboring TADs, is inhibited or prevented by structures that we call TAD boundaries. We still do not know what necessarily defines a TAD boundary and what is required to give rise to a TAD boundary. But we do know that there are certain epigenetic features that are associated with TAD boundaries. The most commonly known is probably the CTCF binding or cohesin and other proteins associated with it that are seen at TAD boundaries. There are also enrichment of certain activating histone marks, as well as depletion of certain histone marks, mostly the silencing marks. So while there still remains a lot to be known about TAD boundaries, what is pretty clear is that these boundaries are important in maintaining gene regulatory order in the genome. In fact, removal or disruption of boundaries can result in ectopic interactions. Ectopic interactions. For example, in the schematic view here, you can see enhancer that's supposed to only interact with gene 1. After removal of the boundary, can now interact with gene 2 that is normally silenced, but now is expressing. And there are several diseases now that have been described as being a result of disruption of TAD boundaries, including these limb malformations that I'm showing you here. So given the importance of TAD boundary and TAD organization in general in gene regulation, it seems logical to study this aspect when it comes to understanding species evolution. However, for a very long time, TADs were just assumed to have remained conserved across evolution. We are, however, now realizing that that may not be the case, and that's beautifully argued and described in a commentary by Ayres and Gilad quite recently. If you're interested, you can check that out. It's very nicely written. In fact, just like any other functional aspect of the genome, we may expect that highly conserved aspects of TAD organization likely reflect critical mechanisms in genes, and TAD rearrangements in TADs happening in a lineage-specific fashion may explain species-specific traits. For example, if we see some TAD organization changing specifically in the humans, that might be related to emergence of a human-specific trait. So that's kind of what we are interested in and what we wanted to study. In order to study TADs and predict TAD boundary placement, we use a technique called Hi-C. I don't want to bore you with the details, but Hi-C generally is a technique based on cross-linking the DNA. You cut the DNA with a restriction enzyme, then you fill it in with biotin labeled marks, nucleotides, and then you shear your DNA further, you pull the ligation regions down, and you end up with a library that contains fragments, hybrid fragments from different regions of the genome that were interaction in 3D space. You can sequence these libraries and process them, and oftentimes the process files at some point of time will look something like this, where you have heat maps showing frequency of interaction between pairs of regions, pairs of loci. The data is often presented either in this square shape or in a triangle format. They're equivalent. The square is basically just a mirror of the triangle at the diagonal. Each of the cells here represent interaction between pairs of loci, and the color indicates intensity or frequency of the interaction. So for example, this square here represents frequency of interaction between this locus, and this locus. So that gives you a measure of interaction. So in our study, we, in order to investigate conservation of TADs across mammalian species and try to study their functional implications, we selected 4 primate species and 4 rodent species. And we collected liver or lymphoblastoid cell lines due to unavailability of liver tissue from the endangered gibbons. We had 1 male and 1 female replicate. We generated Hi-C and ChIP-seq data from all these individuals and integrated the data along with some genomic data to characterize TAD boundaries within each species. We then compared TAD boundary presence and absence across species in order to classify TAD boundaries into evolutionary groups. So we had ultra-conserved boundaries that were present across all species. We also defined human-specific boundaries, as you can see schematically here shown, boundaries that were present in human but absent in all other species, and so forth. We have primate conserved, mouse-specific, and rodent conserved. We we were surprised that despite the common notion being that conservation, the boundaries are quite conserved during evolution, we found that less than 20% of the boundaries in both the human and the mouse genome were ultra-conserved, were conserved across all species. And a pretty similar proportion of the boundaries were actually specific to the species in question. So that was quite interesting to us. We next wanted to see if these boundaries that are so very different in their conservation level also differed in their genetic and epigenetic features. Of course, a boundary is known to provide insulation, so we wanted to see if their insulation was any different. And in fact, it was, otherwise I wouldn't be presenting it to you today. We found that the more conserved boundaries are, the higher the insulation strength was overall. If you overlay the interaction frequencies of all ultra-conserved, all primate-conserved, and human-specific, and use the same for the mouse, you can see, and this center here represents the TAD. This is distance to TAD, so 0 is at the TAD. You can see overlaying the signal across all these boundaries show that there is a lot less interaction, and that's indicated by the darker blue shade in the ultra-conserved or more conserved boundaries than there is in the lineage-specific ones. We also found higher enrichment of active chromatin states or open chromatin states at the more conserved boundaries compared to the lineage-specific ones. And we found more CTCF binding sites in the ultra-conserved boundaries compared to the lineage-specific ones. We also investigated transposable element content of these boundaries. If you're not familiar with transposable elements, these are also known as jumping genes. They're selfish repetitive DNA fragments that can change their position in the genome and or propagate in the genome. They were previously ironically known as junk DNA, but we now know that they can be co-opted in the genome to provide various advantageous function. One of which is that has recently come to light is that they can actually provide CTCF binding sites and function as TAD boundaries in the genome, or give rise to TAD boundaries. So that's quite interesting. And that's the main reason why we wanted to investigate them at these different evolutionary groups. To make a long story short, what we did observe was that there was an overall agreement between the level of boundary conservation and the age and conservation of the TAD boundaries of the TE families found enriched at the CTCF sites of these TAD boundaries. So I want to just point you to these two examples here. So the mirror elements, these are one of the most ancient transposable elements that are found in the mammalian genome. They were enriched in the more conserved boundary groups, while in the lineage-specific or the species-specific TAD boundaries in both human and mouse, we found the B elements, and in human we found the ERVs. These are some of the most recently active transposable elements in the genomes of mouse and human respectively. So this agreement between transposable element age and estimated TAD boundary age indicates that TAD transposable elements have shaped both conserved and lineage-specific genome conformation. And to provide a couple of examples here, on the left, we have an ultra-conserved boundary. You can see there are several CTCF binding sites at this boundary. One of them, if you zoom in on it here at the bottom, the repeats are shown here at the top. You can see that there's a mirror element providing, likely providing this CTCF site. It matches perfectly with the peak. A human-specific boundary on the right is shown here. It contains one CTCF site, binding site, and that binding site appears to be provided by an ERV element called LTR13. So overall, not only did we see high diversity in the level of conservation of TAD boundaries, both in the human and mouse genome, but these boundaries also seem to differ in their genetic and epigenetic properties. But what about their function? Does their conservation reflect any differences in their function, or anything about their function, really? Well, the easiest way to do that and to look into this is to just do gene ontology enrichment for these ultra-conserved boundaries. And what we found was that the ultra-conserved boundaries were, in fact, and the genes near ultra-conserved boundaries were enriched in functions implicated in early development of anatomy. And obviously, these processes are highly crucial for an organism's survival and fitness. So in order to test their functionality, we performed CRISPR-Cas9 in mouse models. We selected 2 candidate ultra-conserved boundaries. I'm only going to present one of them today in the interest of time, but we have a preprint out and bioRxiv that you're more than welcome to check out if you want to learn about the other model as well. After generating these models, we performed molecular and morphological phenotyping. So this mouse that I'm going to tell you about was generated by Wei Li, a postdoc in Nadav Ahituv's lab at UCSF, one of our great collaborators throughout the years. You can see this ultra-conserved boundary was located upstream of the MICE2 gene. MICE2 is a transcription factor that's highly implicated in numerous tissues, so it really is functional everywhere. But we focused on the heart as it's been widely implicated in heart development. As you can see, there's a CTCF binding site here overlapping the boundary, and we were able to confirm that our CRISPR deletion removes this CTCF binding site. Using high-C on heart tissue from knockout and wild type, we were able to show that you can, I mean, this is overlaid, so you can see this is where the ultra-conserved boundary is. And the ultra-conserved, you can see in the wild type liver and heart, you can see that the boundary is still there. In the knockout heart, however, you can see that the removal of the boundary really results in an increase of interaction indicated by this, the increase in red here between the 2 neighboring tabs. You can also see it quite nicely in the insulation curve here. Insulation curves are an indication of the frequency of interaction. We often see a dip at TAD boundaries. So you can see the wild type in green has a little dip here at the TAD boundary, ultra-conserved TAD boundary that is absent in the knockout. When looking at gene expression, looking at using qPCR, we also see that MICE2 is highly overexpressed in knockout mice compared to the wild type. And we were lucky enough to have really great collaborators here at OHSU that were willing and able to look at the heart morphology for us. And here are images of 2 examples of a wild type and a knockout mouse. And what they noticed was that there was more compaction of tissue in the knockout. You may be able to appreciate that even in these images. The tissue just appears a little denser. There's also some abnormal trabeculation and knockouts. While it's a little hard to appreciate in these images, the knockout hearts were more elongated as well. These mice were quite young when we took the hearts. They're post-natal day 5, but it is possible that these phenotypes will result in cardiomyopathy later in life. So this ultra-conserved boundary, at least, seems to be functional, and removing it results in significant phenotypes, both at the molecular level and morphological level, which is consistent with our hypothesis. What about the human-specific TAD boundaries? Can they provide any insight into human evolution? Well, again, gene ontology enrichment to the rescue. We found enrichment of significant enrichment of genes implicated in synapse assembly. And as you know, the human brain is probably one of the most well-known features that sets humans apart from other organisms. And synapsing really sits at the center of these differences. Synapse timing is longer, and during development of humans, human neurons form more synapses than other primates. And the morphology of synapses are different in human compared to other organisms. So this really is a differentiating feature in humans compared to other animals. And we also found that many of the human-specific boundaries were also associated with genes implicated in human neurodevelopmental disorders such as autism. So it is possible that disruption of these boundaries contributes to these kinds of disorders as well. So in order to investigate their functionality, again, we used CRISPR-Cas9 in human cell lines. So this candidate boundary that we selected was located upstream of OTS2 gene. The the cell lines that were generated as part of this investigation were generated by Lana Harshman in the Nadav Ahituv's lab at UCSF. And so she generated these deletions in iPSC, human iPSC cell lines. We selected this boundary because OTS2 is a very interesting gene. It's involved in multiple neurodevelopmental processes, including corticogenesis, neuronal migration, and neurogenesis. It is also implicated in many different neurological diseases including autism, and it's also shown to contain the most significantly accelerated region that differentiates human sequences from other primates, including Neanderthals. So it's both implicated in human disease as well as human evolution. So Lana deleted this boundary in special human iPSC cell lines that have been engineered to be easily differentiated to neurons. And she sorted them, grew them, and differentiated them into human neurons. And using Hi-C, I'm just showing you the insulation score here. What you can see is that there's a divergence in the insulation at the OTS2 locus downstream of the deletion, where the wild types have less insulation. And we also consistently see higher expression of the knockouts at the OTS2 locus. So disruption of this boundary, this human-specific boundary, led to not only to changes in the chromatin conformation, but also led to significant changes in expression in human neurons. So overall, what we found is that in contrast to the common belief that TAD organization is conserved across evolutionary time, we actually found quite high variation in the level of conservation of TADs across different species. These different evolutionary groups or conservation groups had different genetic and epigenetic features, there was also, just like any other functional element in the genome, there was an association between the level of conservation and their function. So we saw ultra-conserved boundaries often being associated with important developmental genes, and we were able to confirm functionality of these boundaries using CRISPR-Cas9. On the other end of the spectrum, the human-specific boundaries, at least based on that knockout that we did, seem to be functional, and they may have contributed to shaping human-specific features, such as the human brain. With that, I would like to thank past and present members of the Carbona Lab, especially those that contributed directly to this work. Most are bolded here, and also our collaborators within and outside of OHSU, the given conservation resources, as well as funding sources. And as I mentioned, we have a preprint out in bioRxiv. There's a lot more to the story that I did not present in the interest of time, so you're more than welcome to check it out. And thank you.
Thanks, Mariam, for this great talk. And I really encourage you all to go through the bioRxiv preprint, because it was really amazing when I first saw it. So we only have time for one question, and I will have this question from Q&A box by Irene Kaplow. Irene asks, do more conserved TAD boundaries tend to have higher nucleotide level conservation, for example, higher phyloP scores? And are there conserved TAD boundaries with low moderate nucleotide level conservation scores?
That's a really good question. So I haven't looked specifically at a single nucleotide level, but we have looked at enrichment of highly conserved fragments of the genome. We did see higher conservation of the sequences in the ultra-conserved boundaries. However, one thing that I did not present is that the ultra-conserved boundaries are also, they overlap genes more often. And it's really difficult to tease apart if this higher conservation of sequences is just due to the fact that they are overlapping genes more often, because coding sequences are by definition more conserved. So there is, yes, to answer your question, yes, there is more sequence conservation at these regions, at these loci, but there also is this caveat of other things that are also associated with the ultra-conserved boundaries. It's hard to differentiate if there's a difference. I haven't looked at individual boundaries to see if there are specific ones that are highly variable in sequence. One thing that I would say, though, and this is based on investigating many different genetic and epigenetic aspects, is that it does seem in general that it's more so that the function is conserved and not the sequence. So it seems like even it may not be the same. So if a boundary is present in both human and mouse, and there's a CTCF site, for example, provided by a transposable element in human, it may not be the same transposable element in the mouse that is providing the CTCF binding site. So the CTCF site is there, but the mechanism that is given rise to that function could be variable across species. And that's something that was really interesting to me and surprising to me as well. So sequence conservation is there, but I don't think that's, I don't think you necessarily need sequence conservation to have a conserved boundary.
Okay, thank you from Mariam for this amazing presentation and for this amazing question by Irene. Now we will move to our second speaker, and Matteo will introduce him.